Descriptions of behavior patterns. Clibanarius tricolor, Gibbs, 1850. Family Diogenidae. This species is discussed first because of the more extensive investigation of its behavior. Descriptions of the methods used for most measurements will be given in this section but not repeated for the other species. This small active crab is certainly the most abundant and widespread hermit crab in the waters around Curaçao. Its abundance makes the fact that it has not been reported from the island seem unusual. The blue, orange, and yellow-banded ambulatory legs are strikingly colored. As with most species, the walking legs are almost all that an observer sees in the field. It is a small crab, large males attaining a cephalothorax length, cl, of only 10 millimeters. Most individuals are less than 5 millimeters in cephalothorax length. Ovigerous females with cephalothorax lengths of 2.5 millimeters have been collected. Males are generally larger than females, as was true with all the hermit crabs studied. Other than general size, there is no visible external sexual dimorphism. Bush, 1930, and Perez, 1932, have reported minor sexual differences in 2 European species of hermits. But these would be difficult to use for sex identification in the field. Populations of Clibanarius tricolor were found in most of the areas inhabited by marine hermit crabs. It was very abundant in shallow waters, along open shores on both the north and south coast of Curaçao. It was not found below the very rough, surf-pounded cliffs on the north shore, but was in tide pools filled by spray from the breakers. It was reasonably numerous in those bocas and bays where the surf action was reduced. It was rarely located below 3 meters of water and was most abundant in 1 meter or less. In addition to open water, rock-covered areas, tricolor was present in the entrances to bays such as Paguridae and Saint Martha Bay. It was also found, although not abundant, on some of the mudflats of Paguridae Bay and Spance Water, but appeared to be absent from many of the mudflats near Bracaput. The micro-distribution of individual groups varied according to local ecological conditions. In all areas, this species is nocturnal. During the day, the crabs move about slowly or rest. Feeding activity begins at dusk and continues throughout the night. In certain areas, the members of a population are very contagiously distributed during the day. A gentle slope, rocky bottom, and open circulation seem to be conditions that facilitate the formation of these dense aggregations. The crabs congregate among small rocks in about 30 centimeters of water. In areas where the water circulation was not as good, such as inside Paguridae Bay, or the bottom contained large rather than small detritus-covered rocks, North Shore bocas. The crabs never formed the very dense groups such as were found in front of Carmabi, outside of Saint Martha Bay, and on the shallow shelf of Boca Saint Michel. In some areas, the number of individuals was systematically counted. A half-meter square was used to mark off quadrates, and all hermit crabs present in each quadrate were counted. In some cases, the sampling was carried out along a line, parallel to shore, since Clibanarius tricolor was absent elsewhere. Usually an area was chosen which included a concentration of crabs, but the limits of the total area measured were defined largely by the author's fortitude. In some cases, The crabs in a quadrate were collected and later counted in the laboratory. The distributions were then tested statistically for non-randomness. The index of dispersion, IM, introduced by Marista, 1959, was used. If this index is equal to 1, the individuals within the sample are randomly distributed. If IM is greater than 1, the distribution is contagious. Marista also gave a formula for calculating an F0 value to determine the statistical significance of the IM. Tables 1 and 2 give the results for C tricolor of the measurements taken in different areas. It can be seen that in all cases, the IM value for the distribution is much above 1, and the F0 value shows that the value is statistically very significant. P is less than or equal to 0.01 in all cases. In some cases, such as St. Marta, IM equals 1705.2, and Carmabi, not equal to 2, IM equals 2865.81. The F0 value was not fully calculated. A total of 53.5 square meters was sampled. The density of tricolor in the areas sampled ranged from 0 to 928 specimens square meters. Table 1. Field distributions of Clibanarius tricolor. A table we are not reading. A field count taken early in the summer of 1962 could not be listed with the other measurements due to the method used. In this measurement, a group of Clibanarius tricolor was collected and counted. The group occupied an area 10 centimeters wide and 30 centimeters long. In the surrounding 16 square meters, there were 175 individuals of tricolor. In the group, there were 3,697 tricolor, 5 empty gastropod shells, and no other species of hermit crabs. This distribution corresponds, in order of magnitude, with the dense aggregations at St. Martha and Carmabi. In all the measurements of field distributions, the index of dispersion indicates that this species distribution is strongly contagious. In the very dense aggregations, individuals are often congregated 4, 5, or more deep on top of one another. These aggregations of Clibanarius tricolor are usually formed in relatively open areas, spread over several of the medium-small rocks which cover the bottom. Several aggregations were located at least partially beneath the overhang of large rocks. The individuals concentrated in these groups were mostly small and medium-sized individuals of less than 4 millimeters cephalothorax length. Larger individuals, mostly males, were present about the perimeter of the aggregation and adjacent area. At about 16 hours, the crabs in the aggregation began moving about within the limits of the group. When a crab came to the edge of the group, it turned and moved back into the group, but by 16:30 hours, the top layers of the aggregation were moving out in the direction of the nocturnal feeding area, and by 1700 hours, all individuals had left the area of the diurnal aggregation and dispersed over the surrounding rocks for several square meters. Members of a group all move in one general direction. In one section of the shallow waters in front of Carmabi, there were several aggregations just a few meters from one another. In the evening, members of one group moved into deeper water, almost directly southwest. They eventually spread out over a semicircular oval area about 4 meters in diameter, southwest from the location of the diurnal aggregation location. The second aggregation in the area mentioned also started moving out at 1630 hours and fed during the evening and night. However, they moved parallel to and slightly toward the shore, i.e. west and west-northwest. As the crabs move outward and throughout the night, they move their chelipeds up and down from the substrate to their mouth regions, picking at the detritus-covered rocks on the bottom. Table 2. Field distributions of Clibanarius tricolor, which we are not reading. A pilot experiment was carried out with these aggregations to measure group stability. Several hundred individuals of one group were removed, dried, and marked on the shell with red ink, and several hundred of the other group were marked with blue ink. These crabs were replaced in their respective groups, and daily counts were made thereafter. The ink used did not last for very many days. During the day, crabs frequently pick at the surface of the shells of other individuals with their chelipeds. However, the observations for the first few days revealed relative group stability, although there were some blue-marked crabs found in the red group, and vice versa. However, the majority, 82%, of the marked individuals observed remained in their own aggregation day after day. An effort was also made to gain an idea about the spatial ability of these groups. The locations of 3 aggregations were determined in relation to large rocks in the immediate microenvironment. The group locations were noted on March 15, 1963, and every day thereafter until March 25. The observations were carried out at 1100 hours every day. The groups were 10 to 20 centimeters long and 5 to 10 centimeters wide. On the 16th of March, all 3 groups were in exactly the same location but exhibited minor changes in general shape. On the 17th, the first 2 groups were the same, but the third had shifted 120 centimeters westward. By the 18th, the latter had split into 2 subgroups and no definitive group could be found. The first 2 groups maintained themselves as to numbers, but had shifted about 10 centimeters toward the shore during the 10 days of observation. Their shapes did change slightly from day to day. On April 22nd, one of the groups was chosen, and after the aggregation had emigrated in the evening, the largest stone upon which the crabs had been partially grouped for the previous 2 days was moved 10 centimeters toward shore. When the group was reformed on April 23rd, it was in the same location. The crabs were on the exposed sand where the large rock had been. No crabs were on the rock that previously was covered with several hundred individuals. Chemical orientation experiments described in later section. Chemical orientation experiments described in a later section indicate that individuals of Clibanarius tricolor orient strongly toward conspecific individuals. It is possible that the exact location of aggregation formation is determined initially by movement to a limited general area, then more specifically to the presence of conspecific individuals in the area. The crabs feed during most of the night. About 400 hours they start moving directly toward the area of aggregation. As individuals get closer to the formation area, they move more rapidly until in the area. By 4:30, large numbers of the crabs are in the area, and by 500, the aggregation is formed. The animals move about within the group for some minutes, but soon become quiescent. Observations of the daily timing of the movements were carried out on a number of days throughout my period of study in Curaçao. The evening movement occurred at the same time every day. This was true whether the sun had been out continually, usual condition, intermittently, or the day was continually overcast or even rainy all day. The latter climactic condition occurred once during the 10 months during 1962-1963 I was in Curaçao. 3 aggregations of Clibanarius tricolor were checked that day, and all emigrated at the usual time. At this time of day, the sun is at an angle of about 35 degrees above the horizon. At the beginning of the morning return movement, there is no visible sunlight. By the time the group reformed, there is some light, but the sun itself is not visible. I was interested to learn what sort of dispersal patterns would be formed under more uniform conditions, such as could be established in the laboratory. Supplied with fresh running seawater, hermit crabs adapt well to laboratory conditions. As far as could be judged, their behavior in the laboratory and field were qualitatively the same. One behavior which was not fully manifested in the laboratory, however, was the formation of dense groups. As seen below, the crabs did aggregate, but even under the most natural conditions established in the laboratory, the formation and daily dispersal of aggregations did not clearly occur. The first attempt to measure dispersion under more uniform conditions was carried out using small rectangular glass aquaria, 21 by 35 centimeters. The bottoms were evenly covered with fresh clean sand, and the lighting was by overhead lights about 2 meters above the level of the tanks. The natural light cycle was not altered. Counts of the individuals in 15 quadrates, 7 by 7 centimeters, were taken at various hours on 2 different populations established from portions of aggregations collected in front of Carmabi. The results of these counts are given in Table 3, along with the index of dispersion and F0 values for each distribution. As can be seen, especially in population not equal 1, there was a tendency to group in the corners and along the edges of the tank. In order to avoid this tendency and present a more uniform environment in which the crabs could disperse, a circular container, 38 centimeters in diameter, was used. Fresh seawater was introduced into the center of the container. A uniform sand bottom was present. The top of the container was divided into 32 equal segments by radii. Counts were made of the individuals of 3 populations established in this container. The results for one experimental population are presented in the histograms of Figure 10. As can be seen, the crabs were distributed contagiously in all cases. 3 additional accounts of the dispersal of the other populations gave similar results. Average index of dispersion 1.37, F0 equals 6.89. These results indicate that Clibanarius tricolor is gregarious in its distribution, since they group contagiously even in a uniform environment. In addition to the previously mentioned marking experiments, some pilot studies on individual daily movements were made. There are populations of Clibanarius tricolor along the east shore of the entrance to Biscadera Bay. The bottom drops quickly to about 3 meters after a shallow shelf about 50 centimeters wide, and tricolor is found mainly on this shelf. In 3 separate experiments, a total of 65 individuals was marked and placed back in the water. In each experiment, the area was searched for marked crabs for 5 to 12 days after marking and release. As in other field marking experiments, recovery of marked individuals was not high. Figure 11 shows the distances which marked crabs moved from day to day. All measurements were made around 1200 hours. A total of 80 individual daily movements were measured. The mode of the results is the 0 to 20 centimeter movement group with 16 instances. The next largest is the 4-meter group with 10 cases. The greatest distance recorded for a crab's movement in one day was 10 meters. Table 3. Laboratory dispersal of Clibanarius tricolor. A table we are not reading. Figure 10. Laboratory distribution of Clibanarius tricolor. Histograms of number of individuals found in 32 equal segments of circular container. Figure 11. Daily movement of Clibanarius tricolor. Distances moved in 24 hours by individual crabs. Clibanarius tricolor is neither a rapidly nor slowly moving crab relative to other species. As in most hermit crabs, the gastropod shell is dragged along on the substrate. Although surprisingly agile for an organism carrying so much extraneous weight, individuals often tumble over backwards when attempting to crawl over an obstacle and lose their hold on it. They are usually able to right themselves rapidly through movements of their second ambulatories. All walking and crawling movements are carried out by the ambulatory legs, the second and third periopods. The chelipeds in this species are small and weak and rarely used for support. In feeding, the chelipeds are used alternately, picking up bits of detritus matter or occasionally larger food, fellow hermit crabs, dead fish, or mollusks. Clibanarius tricolor has in its behavioral repertoire most of the behaviors mentioned in the introduction to this section, although some are not strongly expressed. When two individuals come into contact, they often show no change in their behavior, and one may crawl over the other, or they may pass one another without any apparent notice or contact. Even when aggressive movements are made by one crab, the other frequently shows no change in behavior. Such a manner of interaction is logical in a species that lives in such continual intraspecific contact. When two crabs come in contact, the most frequently used aggressive signal is the single ambulatory raise. The movement can be executed by the crab when it is on a level substratum, or while crawling about on objects in its environment. When walking in a normal manner, the ambulatory legs are held at an angle of about 25 degrees from the dorsal-ventral plane of the animal. The ambulatory raise is executed with the limb held rigid and straight. Moving through about 60 degrees, the limb is brought almost perpendicular to the dorsal ventral axis. However, the leg is rarely raised to a completely perpendicular position. In the raised attitude, the distal segment is very slightly inclined downwardly. The upward motion takes from 0.18 to 0.24 seconds. All speeds measured from motion picture films. Once in the raised position, the leg may be immediately lowered, i.e., no frames of film in which the limb is clearly maintained in the extended position, held in place for a short time, or be held in place for some seconds. This maintaining of a sustained raise was rather common during aggressive interactions between crabs of equal size. The downward movement is not as stereotyped as the upward motion. The limb is not always maintained in a rigid manner, and the distal segments may be moved before the more proximal. The speed of movement is highly variable. The leg may be brought down rapidly, 0.24 seconds, but the process is usually slow, over 0.5 seconds. A second raise may be executed without returning the limb to the neutral resting position. The forward component of the ambulatory raise is slight in Clibanarius tricolor. Both of the second periopods may be raised as described for the single raise. Usually both are lifted simultaneously. However, occasionally the movement of one limb is started before that of the other. This asynchrony occurs more often when the execution of a double raise quickly follows a single ambulatory raise, and the limb involved in the latter behavior is not returned completely. In more intense aggressive interactions, individuals of tricolor may employ all 4 ambulatory legs in a quadruple raise. While all 4 legs are engaged in this manner, the animal maintains an upright position by using its chelipeds for support and/or leaning back, balancing itself on its shell. The posterior legs are not raised as fully as the second periopods. They usually are not lifted above 75 degrees from the dorsal ventral axis. The cheliped extension behavior pattern is not strongly developed as a symbolic aggressive movement in Clibanarius tricolor. Often this movement of the chelipeds is interrupted in its motion by striking against the shell of another crab. Although the behavior pattern appears to have communicative value, its execution as to speed and position is variable. The chelipeds may be raised rapidly or rather slowly to various positions between perpendicular to resting position and parallel to the substratum. However, quite frequently, a crab does execute a rapid movement, less than 0.5 seconds, bringing one or both of its chelipeds through about 80 degrees arc to a position slightly below parallel to the substratum. On a few occasions, I observed individuals leaning back on their shells execute a sustained quadruple ambulatory raise and a double cheliped extension at the same time. As mentioned above, the behavior of one individual may appear unchanged by social contact with a conspecific, even when one organism executes an aggressive movement. However, the performance of an aggressive movement usually is immediately followed by 1. An aggressive movement by the other crab, 2. Some form of retreat by the other crab, of 3. Further aggression or retreat by the original individual. Long series of aggressive exchanges do occur, and equally matched pairs may interact for some minutes. In long series, the most common situation is that the 2 crabs, facing one another about 5 millimeters apart, both execute sustained single or double ambulatory raises, holding the limbs in the raised position for many seconds. Often, one crab reverses or alters its direction of movement, clearly in response to the approach of another crab, before any aggressive movements are made. In addition to backing away and/or changing its direction of forward movement, a retreating crab may duck back inside its shell. Sometimes the other crab will turn the shell of a retreating crab over on its back. It may examine the whole of the shell with its ambulatories and chelipeds, or pass on once it has turned the shell on its back. This behavior was more frequently executed following a long interaction in which one crab finally retreated, moved back, and was then grasped by the ambulatories of the other crab and turned over. An individual which has been on its back, either by accident of the actions of another crab, always comes to the entrance of its shell aperture in the same position. The chelipeds are held at an angle of about 45 degrees to the anterior-posterior axis. The ambulatory legs are held very close together at first, but as the crab comes out of the shell, they separate slightly. Both pairs of legs are widely spread, approximately in the position of a quadruple ambulatory raise. The crab may maintain this posture for several seconds before righting itself through movements of its ambulatories. Clibanarius tricolor was the first species in which I observed shell-fighting behavior. As the attacking crab approaches another, an exchange of aggressive movements may take place, or the other crab may retreat right away. An attacking crab often moves into contact from behind and turns the other on its back. After moving into an opposed position, with apertures directly opposite one another, the attacker may then turn the crab and shell over several times through 360 degrees by movements of the ambulatories. Some investigation of the shell aperture and interior by movements of inserted chelipeds and ambulatories may then occur. With the walking legs spread on the outside around the edge of the aperture, approximately forming a horizontal X, the attacker goes through positioning behavior. This behavior pattern is relatively brief in C. tricolor. The body and shell of the attacker move in a horizontal figure 8, with the ambulatories remaining stationary on the defending crab's shell. The motion may be strong enough to rock the defending shell from side to side on the substrate. Each cycle of movement takes about 1 second, and usually 3 to 5 cycles are executed before wrapping begins. Initially, wrapping is always carried out with all 4 legs outside, but in prolonged fights, 1, 2, or all ambulatories may be inserted inside the hole of the defending crab's shell. In wrapping, the ambulatories and cephalothorax remain approximately stationary as the attacker's shell is raised until about half of the cephalothorax is exposed. The shell is then brought down rapidly. The downward motion brings the upper edge of the shell very close to the upper edge of the defending crab's shell. The area of the first whirl of the shell just below the rim of the aperture of the attacking crab comes in contact with the corresponding position of the defending crab's shell. This contact appears to produce the sounds sometimes heard accompanying the wrapping motions. The motion is somewhat circular when viewed from the side, moving about 30 degrees through an axis running parallel to the surface of the shell apertures and located near the lowest portion of the first whirl of the shell of the attacking crab. The complete cycle of movement from up to down and back up averaged 0.33 seconds in duration. The range for complete cycles, measured from films, was 0.18 to 0.62 seconds. The exact speed of downward motion could not be measured since the complete action occurred in less than 0.06 seconds, the shortest period of time measurable with the films taken. The movement from the down position to the raised attitude was 3 to 5 times as long as the downward motion, taking between 0.18 and 0.30 seconds. The attacking crab always began its downward motion as quickly as the shell was fully raised. The time the crab remained in the fully down position was variable. An attacking crab may wrap just once, but more frequently it carries out a series of wrapping movements. The series may vary from a few to 20 to 30. The later wraps in a series almost always are weaker and move through a shorter distance. In addition, a series of wraps may all be weak short movements of less than 1/4 the length of the cephalothorax of the crab. After a series of wrapping movements, a crab may go into the aperture of the defending crab head first. The 2 shells are closely pulled together and neither crab can be seen. This position may be maintained for minutes without any visible motion. More wrapping may begin directly from this position, but more frequently the attacker comes out and begins wrapping either after or without positioning behavior. Between series of wrapping movements, the attacker frequently moves its chelipeds up and down alternately in the shell aperture without going into the hole. A crab can begin a series of wraps with all legs outside, and one by one move all ambulatories into the aperture with very little interruption in wrapping. Although wrapping always follows positioning behavior, the latter is not a necessary prerequisite for the former, and cheliped movements may or may not precede wrapping or positioning. A complete shell fight, successful or unsuccessful, may consist of a minimum of positioning followed by one short series of wrapping motions. Conversely, some unsuccessful attackers may fight for a number of minutes and execute over 100 wraps before releasing the other crab. When it does give up, an attacker almost always leaves the defending crab on its back, and as the defender tries to right itself, it may again be attacked. Occasionally, after a sequence of shell-fighting interaction, the defending crab will come up and, by a series of aggressive movements, cause the attacking crab to retreat. The last behavior of a successful attack invariably is wrapping. After sufficient stimulation, the defending crab starts moving up to the entrance of its shell aperture. As this happens, the attacking crab moves back away from the defender and from a slightly lower position carries out a few more wraps. As the defending crab moves out of its shell completely, the chelipeds of the attacking crab usually rest on the lower lip of the aperture. Occasionally they appear to be in contact with some part of the losing crab, and may actually exert some physical force to hasten or assure the exit of the loser. However, in the majority of fights, no such physical pulling or tugging was observed. After the loser is extracted, the winning crab usually investigated the interior of the shell briefly with its chelipeds and ambulatories, while the new loser moved about on the back of the shell of the attacker or its own shell. In crowded conditions, such as are common in nature for this species, the loser is subject to almost certain death from the attacks of conspecific individuals if the winner prevents its entering a shell for more than a few seconds. A naked crab cannot protect its abdomen satisfactorily, and is torn to bits rapidly. After initial investigation, the attacker usually then moves into the new shell. As it moves, abdomen first, from one shell to the other, it quickly moves its ambulatories from the shell it is moving into to the shell it has just occupied. It does this in such a manner that it maintains possession of both shells, even after it has moved into the new one. The winning crab frequently backs all the way into the new shell, as far as its hold on the old shell will allow, presumably testing the fitness of the new shell. It may change back to the old shell, but normally once won, a new shell is quickly occupied, and the losing crab moves into the old shell of the winning crab. On one occasion I observed a successful fight, complete with double shell change, followed immediately by a second fight between the same two crabs. The winner of the first fight was again victorious, and won back the shell he had just vacated. As in many crustaceans, mating in Clibanarius tricolor apparently can be successful only for a short time following a female molt. Some time prior to her molting, a female is grasped by a male. This time may vary from a few minutes to 5, 6, or more hours. 6 hours was the longest period of time a mating pair was observed prior to copulation. Longer pairings probably do occur. A male may grasp a pre-molt female and after moving the shell around to an opposed position, may hold it quietly for some minutes. More often, the male moves one or both chelipeds down into the aperture of the female and moves them up and down. These movements of the chelipeds occur periodically throughout pre-copulatory activity. The male may then carry out a few rocking movements. These movements are of two types in tricolor and are the most consistent and conspicuous elements of mating behavior. The male holds the female with his ambulatories in a wide X around the aperture of the female. The body of the male is not directly over the female, but the mouth region is over the lower lip of her aperture. The male holds its chelipeds just at the entrance of the shell aperture. In this position, with the female below and slightly in front of the male moves the female back and forth around an axis running through and perpendicular to the plane of her aperture. Initially, the male always performs a gentle rocking movement in which the motion is very slight, the movement of the male ambulatories not exceeding the length of a tricolor eye stalk, less than 1 millimeter. The rocking is carried out in series of 3 to 6 or more cycles at the rate of 2 to 3 per second. The apex of the female shell only moves through about 10 degrees arc during this rocking. Series of gentle rockings may be interspersed with quiet maintenance of position or an alternating movement of the chelipeds up and down in the aperture of the female. Occasionally, the male will push the female back and forth about an axis parallel to the columella axis of the female. This may either precede or follow rocking movements. At first occasionally, and later more frequently, the gentle rocking is interspersed with violent rocking by the male. This is also affected by movement of the male ambulatories, but is more rapid, 0.18 seconds per cycle, and the female is moved through a greater distance, 40 to 45 degrees arc. The movement is not purely circular, but approximately in a horizontal figure 8. That is, the tips of the male ambulatories move asynchronously in ovals rather than in simple arcs. This rocking shaking may proceed with the female shell on the substrata, or the male may lift the shell entirely off of the substrate and rock the female about in the water. The latter can only be done when the male is substantially larger than the female. Males of mating pairs average 47% larger cephalothorax length than females. The range for the pairs measured was 20 to 70%. The violent rocking behavior is also carried out in short series of 3 to 6 cycles. Violent rocking is usually followed by the gentle rocking and or cheliped movements by the male. As this pre-copulatory activity proceeds, the female may occasionally come to the surface of her shell aperture, only to retreat again following cheliped movements by the male. Several hours before copulation, the male may begin violent rocking and the frequency of occurrence of all behaviors gradually increases. Shortly before copulation, the female molts. Non-virgin females usually release the larvae from previous matings before they are grasped by a male. I did find a few males performing early precopulatory behavior with females which were still carrying a matured brood. When the female molts, the male grabs at the old exoskeleton of the female and pulls it out of the shell. On several occasions, the male actually helped the female out of her exoskeleton by actively pulling at the legs and chelipeds. After helping the female out of her old exoskeleton, the male moves his chelipeds up and down in the aperture of the female. After a series of either type of rocking or cheliped movements, the female comes toward the rim of her shell aperture, and as she eases out, she moves her chelipeds and antennules rapidly in the mouth eye stalk region of the male. When the female performs these movements, the male begins to ease out of his shell. As he moves out, the female does likewise, keeping her antennular flagellae near the mouth region of the male. The body of the female is completely covered by that of the male in the copulatory position. The cephalothorax of the female does not come completely out of her shell, but just far enough to expose the genital openings on the coxopodite of her third periopods. The male comes further out of his shell, exposing almost the full length of his cephalothorax. By stretching, the male is able to bring the genital openings on the coxopodite of his fifth periopods near the female orifices. The distal segments of the male's fifth periopods move rapidly back and forth between the genital orifices during copulation and may aid in the transfer or placement of the spermatophore filaments. The actual copulation lasts between 1 and 5 seconds. It is terminated as both animals pull back into their shells rapidly. The female pulls completely down into her shell, and the male back to the opposed position, holding the female. It is not known which partner first terminates copulation. After copulation, the male may push the female away and mating is completed. However, that is rare, and a period of post-copulatory behavior usually follows. This is a series of violent and gentle rocking and cheliped movements by the male which appear indistinguishable from pre-copulatory behavior patterns. There may be some differences between pre- and post-copulatory movements, but none were detected in the matings that I could follow to completion. Post-copulatory behavior may be carried out by the male for up to an hour. Just prior to termination, the female comes up and again moves her chelipeds and antennules in the mouth-eye stalk region of the male. The male then backs away and the pair separates. The male usually leaves the female on her back and may exhibit some aggression towards her as she attempts to right herself. If a female Clibanarius tricolor is examined just after copulation, one finds her exoskeleton is very soft and her abdomen externally void of eggs. However, new eggs are visible in her abdomen. If a male carried out only a short period of post-copulatory behavior, this will also be the condition of the female when the pair separates. When a pair is uninterrupted and carries out a complete sequence of post-copulatory behaviors for some minutes, at the termination of this sequence, the female possesses a noticeably harder exoskeleton, and most or all of the new eggs are in place on the pelepods. Whether the post-copulatory behavior of the male actually aids the female or simply affords her protection while she is getting the new eggs in place and her exoskeleton is hardening, is not known. If the force of gravity is used in getting the eggs into position, as suggested by the observations of egg placement in Paguridae by Matthews, 1959, the upside-down position in which the female is kept could be an aid to egg placement. As will be shown later, recently molted individuals are more subject to attack by shell-seeking conspecific individuals and are more apt to lose their shell in a fight. Thus, post-copulatory behavior by the male may protect the female from attack by other hermit crabs. A few multiple copulations by females have been observed, but appeared to be rare. Males have been observed to mate with 3 and 4 different females in one day. In a number of pilot experiments, mating pairs of Clibanarius tricolor were separated and the male and female placed in a small dish. In very few cases did the male show any orienting preference toward the ripe female over the control male or female in the dish. When a male did come into physical contact with a ripe female, he usually grasped her immediately, but often a male would pass within a centimeter of a female and show no change in behavior. When a male did orient directly over some distance, it was immediately upon placement in the observation dish, and presumably was visually directed. Experiments using ripe females and control animals contained in small pieces of cloth and offered to males also failed to support the idea of any distant chemical orientation by the male. In one case, a male did pick up the cloth containing a ripe female after wandering into contact, and held it in his ambulatories for several minutes before moving on. Considering the dense aggregation of individuals in which a ripe female would be located, the absence of distance chemical orientation may not be unreasonable. From casual observations, the presence of a lunar cycle in the mating activity of some species was suggested. It was reasoned that since ovulation and mating are closely associated, the presence of new eggs and thus the size of the eggs carried by females would show the presence of such a cycle. Collections of 20 to 25 crabs were made every 3 days during the month of July, 1963. Later, the egg diameters of the females of these collections were measured using an A.O. Spencer dissecting microscope with eyepiece scale. The results are shown in Figure 12. On July 1st and 10th, only 50% of the females were carrying eggs externally. The percentage was higher on most other collection days. Although the limited data gathered does not negate the existence of a lunar cycle and possibly supports the idea, the gradual increase in egg size after a full moon peak and decrease, it is not definite evidence for such a phenomenon. Figure 12. Variation in average egg diameter in Clibanarius tricolor during May 1963. Clibanarius tricolor is a very useful animal for observational work on social behavior. It is common in Curaçao and most of the Caribbean area, and its ecological limitation to shallow water makes field work practicable. Moreover, it shows all the aspects of social interaction observed in hermit crabs. Aggressive interactions occur continuously in a large group. In an aggregation, such as described above, observations of shell fighting and sexual behavior are readily carried out. In a group of about 3,000 crabs, it is usually possible to see at least one pair of crabs in sexual activities in less than 15 minutes of observation. Shell fighting appeared less frequently, but 15 to 30 minutes of careful observation usually allowed detection of one or more shell fights. Sexual behavior was seen 36 times and shell fighting interactions 11 times during the observation of 2,000 interactions in this species. Sexual behavior and shell fights occurred both night and day, although, as might be expected from the marked diurnal grouping, interactions tended to occur more in the day. In order to measure the diel variation of activity in certain aspects of the behavior of Clibanarius tricolor, populations of 20 individuals were established in small glass aquaria in the laboratory. The aquaria were marked on the outside with lines at 3-centimeter intervals. No food was placed in the tanks for the duration of the observations. The animals were observed during half-hour periods throughout the 24 hours. 2 sets of crabs were observed, March 11 and April 4. The number of times any crab crossed one of the lines was recorded, and the total crossings of the members of the group was used as a measure of the level of general locomotor activity. The number of crabs moving their chelipeds up and down, as if feeding, were recorded during each minute of observation. The total number of crab minutes was used as an indication of the level of feeding activity in each half-hour period. As a measure of the level of aggressive behavior, the number of aggressive displays executed by members of the group under observation was counted during each period. Observations at night were carried out with a red light for illumination. Observations in the field and laboratory indicated that white light sources, when covered with heavy red cellophane, were sufficiently red to allow observation of the organisms without disturbing their behavior patterns. Figure 13 shows the results of the diel observations. As would be predicted from field observations, Clibanarius tricolor is relatively quiescent during the day and feeds more intensively at night. Figure 13. Dial variation in activities of Clibanarius tricolor. Average of 2 days measurement. Relative levels of locomotor and feeding activity show nocturnal pattern. Aggressive activity shows evening peak.